Good morning, everybody. Is everybody ready to start today? Okie doke. So today we're going to move on to talk about functional MRI. So just to remind you, so far that what we've done is we've looked at a little bit of history of neuroscience. We've talked about the brain from a cellular level up until an anatomical level. Then we looked at different functional areas within the brain, and we looked at a history of neuroimaging and some techniques that are used for structural imaging and functional. And today we're going to move on to functional MRI. So hopefully this lecture will be a little less intense than yesterday's lecture. Okay, we're going to go over some different parts of functional MRI. And sort of in the same pattern that we've been doing with the other methods, we're going to talk about an overview of it in a brief history. We're going to talk about how it works and the basic principles of why it works. We're going to talk about how experiments are usually designed, how to analyze the data, group analysis, limitations, and problems. So we may not get all of this done today. This is broken down into a couple of lectures. But we'll get as far as we can, and then what we don't get done today, we'll continue on with next week. Functional MRI is a really new technique in the grand scheme of things. So it started being developed in the 1990s, so around 1991, which sort of, for you younger people, it might seem like a long time ago, but it seems like it was just the other day. So it's been really fascinating to watch this field grow and develop over the past 20 years or so. And fMRI is based on the principle that we can detect changes in blood flow across functional states within magnetic resonance. And this is due to an effect called the blood oxygen level dependent response, or the BOLD response. So we'll talk about this BOLD response in a bit of detail. The benefit of functional MRI is that we can get pretty good spatial resolution. And initially, when this technique started, we didn't have very good temporal resolution with it. But we're finding that as we can push the MRI machines faster and faster, we can get a bit of temporal resolution. Not on the scale of an EEG, not on something that occurs instantaneously, but within the range of a few seconds. This technique is completely non-invasive, so it doesn't use any shots, any ionizing radiation. We don't need any kind of dye. So one benefit for this technique is that we're allowed to study children with it generally, and we can study different clinical populations that may not be otherwise able to study if we had to use invasive methods. The studies are fairly easy to implement, which makes it ideal for designing studies and recruiting subjects to come in. And there are numerous applications that we can use with this technique. Ogawa was sort of like the father of functional MRI. So in 1990, he observed that this blood oxygen level dependent signal was present because of this T2 star effect, which we talked about a little bit yesterday. And I'm going to show you a little bit more about why that is today. But he noticed that the areas around blood vessels became more visible as oxygen levels decreased in these areas. So he noticed that near the blood vessels, we saw these local changes in the magnetic field, and he brilliantly decided that we could use this effect to our advantage and study function in the brain. So in 1991, Beliveau observed the first functional images using a contrast agent. And of course, today we don't need to use these contrast agents anymore. So in 1992, the first functional images using this BOLD signal were published. I know we've talked quite a bit about regular MRI, so I just want to show here the difference between MRI and functional MRI. Again, as we mentioned before, MRI is for structure. So the signal that we see from MRI is from hydrogen ions that are present in the water in the tissues. And with regular MRI, the functional state that the participant or the subject that you're scanning is in is irrelevant. So the person could be sleeping, they could be watching a movie, they could be doing any number of things in the scanner. And sometimes, in clinical scans, you want to sedate them so that they're less prone to claustrophobia or things like that. In functional MRI, though, we need the person to be awake and doing something. So we're looking for function, not structure. We're measuring not a signal from hydrogen ions, but a bold signal. And functional MRI requires some kind of oscillation between two states, and we'll see why that is later. But you can see here on the screen the extreme difference in the resolution of these images. So in our MRI image, those are typically collected over a longer period of time. So a scan of this type might take about 7 minutes to complete from start to finish, whereas in functional MRI, we also may have a 7-minute scan, but we're collecting bold information and we're collecting it very quickly. So you can see that the resolution drops dramatically in these scans. So it's a trade-off between how quickly do you want to scan and how much resolution do you want to get in your image. So what is typically done, though, when we collect this type of data is we collect a structural scan first. A lot of times it's done at the very beginning of the scan, so that's the first thing that's done, because if the person isn't able to complete the scan for any reason, then at least what they were able to complete will be salvageable, because we'll still have that structural scan. Because at the end of our functional analysis, we like to put these two images together and map the functional data onto their structural data so we can get a better idea where it was locally in the head. Okay, so we're going to talk a little bit about how it works and the basic principles. And to do this, we're going to talk about the blood oxygen level dependent signal, or the bold signal. Okay, so what happens when we think? Well, the neurons fire in our brain in the area that we're using, and when this happens, the metabolic demands in that area increase. So the tissue surrounding it needs more oxygen, needs more glucose, and in response to this, the cerebral blood flow increases in that area. So this is a local change that occurs, and when this blood flow changes, the amount of deoxygenated blood in that area decreases. And you'll see in a minute why we're talking about deoxygenated blood here. But this is the basic general process that happens. And interestingly, we saw that the brain uses about 20% of the energy of the body. The brain is a very high priority organ to the body. So these changes that occur are noticeable and they are rapid. Okay, so oxygenated hemoglobin versus deoxygenated hemoglobin. So we said that the deoxygenated levels of the blood change locally. So oxygenated hemoglobin, or oxyhemoglobin, is diamagnetic. So that means that it doesn't have any magnetic spin in the area where it's present, or the molecule itself doesn't have that. But deoxygenated hemoglobin is paramagnetic. And if you remember, we said in a material that's paramagnetic becomes magnetic if we put it into a magnetic field. So it has an unpaired ion or something that's making it have an a magnetic property in the field. So when the red blood cells pass through the lungs, oxygen binds to them, to the hemoglobin molecules, and these oxygenated blood cells move through the body to the location where delivering oxygen, and once they deliver this oxygen, once it gets there, the oxygen is released to the tissues, and the blood becomes deoxygenated again. So this is a pretty complicated little structure of hemoglobin here. This is a hemoglobin molecule, and you can see those little blue places are the places where iron binds to the hemoglobin. So you know that, you may not know, but iron is bound in the blood to the hemoglobin molecules, and there are 4 places that it can bind. So this is why if the iron isn't bound somewhere, that it has this odd charge to it. So the decreases in the local amount of deoxygen hemoglobin reduce local magnetic field gradients between the blood and the capillary bed. So when this happens, the values of T2, and particularly T2 star, increase locally, and it's T2 also because remember we said that T2 star is a part of T2 in an inhomogeneous field. And so these properties change locally within the tissue as the blood goes from oxygenated to deoxygenated. Okay, so here's a little example. The neurons at the top here are not firing, and some of the blood going through has oxygen bound, some does not. And when the brain activates on the bottom, the neurons activate, those metabolic demands change, and more oxygenated blood is sent to the tissue. So what happens, here's another schematic of this. In a basal state or resting state, we have normal levels of blood flow, normal levels of hemoglobin, and we have a normal MRI signal. So as in an activated state, we have an increase in flow, an increase in cerebral blood volume, and these ratios of oxygenated blood to deoxygenated blood change in the tissue. So that's what we're looking for here, these changes in the ratios between these blood cells in the local area. So under a resting state, for example, here on this plot, you may have a T2 star, like the red line here, but as the ratio of deoxygenated to oxygenated blood changes, it's going to change the very local effect of the magnetic field in that area, which is going to change our T2 star properties very locally. So you might get just a slight change, and you can see here that T2 star during a task would be different than T2 star during a control state. So thankfully for us, that means we can look at these slight changes in the magnetic field in that particular area or region in the brain, and we can note these changes as the person goes from a resting state to a task-dependent state. So these are happening again, very, very small changes and very, very locally with the changing metabolic needs of the tissue in the brain in response to a task. So the bold signal actually is a slow signal. So this effect takes place over a few seconds. So what happens is a stimulus comes up, and we have this initial dip in the bold response, and then as the person is performing this task and the blood flow changes, we have these changes in this bold signal. So the bold signal will rise, and it will fall again after the area goes back to baseline or is no longer being used. So you can see this is about 6 seconds to the peak of the bold signal after an area becomes activated, and to go back down to baseline, it takes about 15 seconds. So this is a very slow response in the body comparatively to a neuron firing. So if we wanted to measure this, this change in our MRI intensity, we would see this effect in sort of following the same pattern that the bold signal follows. So we would see a slight change in our signal intensity, and then after the stimulus goes off, we would see it go back down again. So this is something, again, that we can measure, that we can see by comparing to two different states over time in response to a person doing some kind of a functional task. I'm not really sure why this initially. Oh, I do know why. I'm sorry. Initially, the area is taking in oxygen faster than the blood response goes to that area. So initially, there's a little deficit. So the oxygen is being used and it decreases, and then as the body is sending more blood, it goes back up again. Does that make sense? So it takes the blood flow is a slow response compared to the signal changes and the metabolic needs. So it takes this little time for it to stabilize in that area. Okay, so we have to remember, though, that the bold signal is an indirect measure of neural activity. So we're having this neural event that's occurring, and we're measuring the bold signal. But in between these two, we have this vascular response. So we're not actually looking at a direct measure of neural activity. We're looking at an indirect response to the neural activity. So we have to be careful in the types of conclusions that we infer from this data, because we remember that it's not a direct response. So people have looked more into what is this underlying neural activity, right? So I told you it's changes in the metabolic demands, and that is basically what it is. But people have asked the question, because what happens is our body actually sends an excess amount of oxygen to that area, so it really sends more than we need, and people aren't sure why this big change occurs, and they're not exactly sure what this underlying neural activity is. So we're assuming it's the neurons firing, but people have addressed this question in more detail as we've gone through the years of fMRI. And so they think this underlying neural activity could be from spiking activity, subthreshold activation, synaptic currents. So the question here is the neural firing directly proportional to the changes in blood flow? That's something that people aren't exactly sure, and they're still trying to understand. So does our bold signal increase proportionally to the neural activity, or is it some kind of a nonlinear event that's occurring? And we're still not 100% sure about those things. The take-home message is, again, this is an indirect measure of neural activity. So now that we've seen what the bold signal is, we're going to look at how the signal is actually measured. So when a person comes in for a functional MRI study, they're first screened by a nurse, and this isn't unique to functional MRI. Every time a person comes in for an MRI scan, they're screened, they're asked a whole page or more of questions about the possibility of having any metal in their body. And interestingly, people sometimes actually forget that they had these things occur. There was an example maybe a month or so ago in the US that a woman came in, she was an older woman, to do an MRI scan. It wasn't at our facility, I'm not quite sure where it was, but she had actually forgotten that she had an aneurysm clip in her brain. So she was asked several times and screened several times, and she said, no, I don't have anything. And she went to do the MRI, and because the brain is soft, an MRI clip is not held into place. An aneurysm clip. It's not held firmly into place like something in the bone maybe. And it was actually pulled through her brain when she was doing the MRI, and she ended up passing away from that the next day. So We are very, very careful with our participants. These are research studies. These are not clinical studies. So none of our participants really have this life or death situation where we need to understand what's going on clinically. So we try to be very careful and very aware and ask them over and over again all these questions about possible metal in their body, possible metal in their eyes. Sometimes when people do metal work in a metal shop, little scraps of metal might fly up and get into their eyes, and they may not even know this has happened. But of course, the eye is also a soft tissue. So if they have any type of metal, especially for a metal shop where they may not be sure if it's a ferrous metal or not, we do not scan them. And if it's something that we really want this participant for some reason or another, they're a very valuable subject to us, we'll have them go and have x-rays of their eye orbits before we put them anywhere near the MRI environment, because you don't want that metal pooling through the eye. So we do an extensive screening process. All females that are scanned have to undergo a pregnancy test. There's no known effects of high magnetic field on a pregnancy, but in the United States, the FDA says that any pregnant woman at this point needs to use caution unless it's a life or death situation about going into the MRI machine. So if we have a person that thinks that they might be claustrophobic, rather than spending all this time and money getting this scan set up for them, we do have a simulator. What it is it's an old MRI machine that no longer has the magnet, and we'll put the person in this simulator. It's the exact same MRI, it's the exact same environment that they would be in for the true scan, but they can lay in there for a few minutes and get the feel of the machine. We have a little thing that plays the same noises that they'll hear when they're in the machine, so that they can decide if they feel like they would be able to lay in this scanner for an hour or more. And we did a study where we looked at children, and this was especially important for making sure the children felt comfortable in this environment before we subjected them to the actual scan. Most of the time when we do cognitive tasks, we want to train the person on these tasks before they go into the scanner. So they have to learn to do a certain task, and we'll be looking at some of these tasks later and how they're designed, but it's good to have them know what they're doing when they're in there. So an hour scan for us costs about maybe $600 to $700 per hour. So, that's not a terrible amount for an MRI scan, but when you're looking at scanning 100 or more participants, you want to be sure that your participants are trained and ready to go when you put them in the scanner. You don't want to spend a lot of time wasted, fiddling around with, do you understand this or not? So we have them trained and ready to go when it's time for their MRI scan. This is the environment that they're in for a functional scan. So I want to point out a few different things here. You see the person laying there, and you see they have this little tilt in front of their face. Well, this is a mirror that we use. I see these arrows aren't in the right place, sorry. That mirror should be over to the person's face. So what happens is we use a projector behind the person to project the task, and the person can see it on the mirror as they're laying there. So they basically see this computer screen while they're laying there, and they don't have to turn their head at all to see it. And this is important because we want to try to minimize head motion as much as possible. Typically, you can collect heart rate and respiration if you're interested in doing so. And the little thing that says mirror should be moved over. That should be the respiratory belt. So you can put a pneumatic belt around somebody's chest and get their breathing rate from that, and that doesn't use any metal. They also have EKG electrodes that aren't metal-based, so we can use those as well if we want to measure. Actually, not the EKG, it's the pulse ox in the finger. So we can measure that while the person is in the scanner as well. You can see here the projector In this case, it's shown in front of the person, but it's the same idea. I think since this was created, they've moved the environment around a bit. But the general point is you have a projector that's projecting your task, and the person can see it on the screen. The head coil goes, like we saw yesterday, that's the RF coil. It goes right over the person's head, and actually this mirror sits right there on the head coil. For our studies, we also use an eye tracker because we have sleep-deprived participants, so we want to make sure that we can see that they're still awake, that their eye is open. So that eye tracker is an additional mirror device that sits on the head coil there and projects back into the back of the machine so we can see their eye on a computer monitor as we sit in the control room. Importantly, the person also has an alarm bell in their hand for any cases of panic, if they feel like they're going to be sick, anything where they need to get out of the scanner right away. We try to tell them if you need to ask us a question or something like that, or if you think there's a problem with the equipment, you're not seeing the task correctly, to tell us in between the noises of the scanner. Because when that alarm bell is squeezed, it makes a big loud noise in the scan room, in the control room. It shuts the computer off, so everything stops, which of course is good because if we need to get to the participant fast, that certainly gets our attention. But sometimes people will just squeeze that ball just to see what it does, or if they if they need to ask us a question or anything like that. So we try to tell them, save that for emergencies if there's a reason you need to get out of the machine as fast as possible. So back to how we measure this bold signal. After we get the person all set up in the scanner and comfortable, we usually put blankets on them. It's very cold in the MRI environment because of all those cryogenics that we use to keep the magnet cool. And a lot of computer equipment is around this area, so not directly in the MRI room, but in the room surrounding it. So they like to keep that equipment nice and cool. So let me go back here. So they're usually have a little pillow under their knees and their heads all packed in with pillows, and we give them lots of blankets to keep warm. And I always think this is sort of a paradox to our keeping them awake all night. If we do these sleep deprivation studies, then we get them all cozy and put them in this little scanner and tell them they need to stay awake. So you can imagine that people have a really hard time staying awake in there. Okay, so once we get the person in there, we have them alternate between two cognitive conditions. And we're going to see this a little bit as we talk about the tasks that some of the different tasks that we do in the MRI machine. So an example, a very basic example would be tap your finger versus don't tap your finger. So in this example, we're going to use the finger tapping as our cognitive state, and we're going to use rest as our non-cognitive state, our relaxed states. So those are two states we're going to use. And what we do is, as this person alternates between these two states, we collect these scans rapidly and repeatedly. So a typical TR, or time of repetition, that we talked of yesterday, to use for this is about 2 seconds. So we collect about 32 slices of data in 2 seconds. So 2 seconds would be from when we start at the bottom of the head to when we get back to the bottom of the head again. So it's really quickly just flying through there using that EPI sequence that I showed you yesterday. And I think it's shown again here in a second. But as we're doing this, we know when the data that occurred during the finger tapping is versus when the data that was collected during the rest state. So we know which scans were collected during finger tapping, and we know which scans were collected during the rest state based on the timing of our task. Another thing that they do in functional MRI is that we try to get as small as slices as we can. So what they do as they're collecting it is they collect every other slice, and then they go and they collect every other slice again. So the reason that this is done is because we're collecting so quickly that the tissue has to return to its normal state, get back in alignment with the field, and we don't want the tissue in the previous slice to be influencing our next slice. So they'll oftentimes skip a slice. That's to allow the original slice to get back to baseline as far as the relaxation in the longitudinal and the transverse planes before they collect the data in the slice next to that. Does that make sense? Okay, so as we're collecting between these two states, we're getting all these slices over time, and what we can do then is we can take an average of all the slices taken over our finger tapping versus an average of all the slices taken over our resting state. And you can see here we can calculate both a mean and a standard deviation from those slices. So basically, let's take our biggest time series of data here, and our time series being each slice or each part of the brain collected over time, and let's stack up all the slices that were collected during finger tapping and make an average and a standard deviation of those, and take all our slices that were collected over the rest and take an average and a standard deviation of those. And then we're going to use this statistical information to try to find changes in the intensity due to this bold effect. Now, these changes that we're looking for are very small. So at 1.5 Tesla, we have about 3 to 5% signal change. So these are not big blaring changes that we're looking at. If we look at the time series of the data that as we collected, we cannot visually see where these blood flow changes are going. These have to be done using a statistical technique. So again, we can detect an average images over time or across cognitive conditions, and we can apply statistical testing to find differences in the intensities. And because we have such a small signal change, actually at 3 Teslas, this goes up a little bit, but it's still less than 10%. So what we don't want is our data to be drowned out by noise. We don't want these variations in the signal to be due to noise effects. We want them to be due to our finger tapping, right? So we try to do these techniques before we apply the statistics to try to minimize the variance or the noise from any other unwanted sources. And we'll talk about this a little bit later. So again, this is the echoplanar imaging pulse sequence. What we do is we flip the neurons, and then we quickly collect, okay? So we flip them all, we collect all the slices, then we go back to baseline, we flip them again. So this is a really fast sequence. And again, we can use also spiral imaging. Spiral imaging is also a fast sequence, and remember the benefit of this is that we don't have to turn any sharp corners. So it's easier to let those gradients move smoothly and adjust them more quickly. Typically though, nowadays, we used to use spiral imaging a lot, and a spiral image was a sequence created by one of our physicists or not by one of the equipment managers. So we usually use General Electric's or Siemens scanners, and these often come with pre-made pulse sequences. So you buy the scanner, it comes with a package of pulse sequences, and EPI is a very typical one that's used. So we also have physicists that try to create new pulse sequences all the time. And in the early days of fMRI, we had a physicist that designed this spiral sequence. So we use this often. But now we've bought a commercial scanner, and we use a lot more commercial technology than was available in the early days of fMRI. So this is the reason why most everybody has switched to EPI, because it's a commercially available sequence now and built into our scanners. Okay, so again, we have to remember that when we collect our functional MRI data, Just like when we collect our regular MRI data, this data is collected in K-space. So it's collected in the frequency space. And to actually get to our image, we need to use a 2D Fourier transform. So again, on the left is the K-space, and on the right is an anatomical image. But in the, like I said, we still need to use this with our functional data. So here is just a little schematic of 1, 2, 3, 4, 5. So a 7-slice study. So our slices are across the top in the rows, or across the rows, and our time series is going down in the columns. Okay, so if we blow up a little area, I don't know if you can see this little square here. If we blow that up just a little bit, you can see sort of what the data looks like on a smaller scale. It's big, it's chunky, it's not a nice anatomical brain like we get with the structural scans. So we have limited resolution. We're collecting very fast, so we're trading off our resolution for our time. Okay, so that's how we measure the bold signal. Now we're going to look at how some of the experiments are designed and how these are used to try to get as good a data as we can. Okay, so the types of studies that people have looked at using, the types of cognition people have looked at using fMRI have been motor studies, and I gave you the finger tapping example. People have done eye movement studies. We're going to look at that a little bit. They've done working memory studies. So we have one task that's called the NBAC, where a person sees, for example, a letter, and they'll be shown a series of letters that are flashing, and they're told if it's equivalent to a letter that occurred 2 times ago, then press a button. If it's not, then press another button. So these are some types of working memory tasks that we have. We also have a spatial NBAC task where we show the person blocks in locations, and we say, if this location occurred 1 time ago, you press this button. If it occurred 2 times ago, press this button. So we can get pretty complicated working memory studies with this. We also study inhibition. So there's a task called the go, no go, where you have a person very rapidly press a button. So we have letters that appear on the screen, X's and K's, and we say, if you see an X, press your finger. If you see a K, don't press your finger. So this, you think, well, that's a pretty easy task, but they occur really fast, and you get so used to pressing that you see the K and you press and you say, ah, I didn't mean to do that. So that's all part of the task. Then we want to see what it looks like when you're thinking, oh no, I messed up. So that's also a cognitive state that we're interested in looking at when we look at these inhibition studies. So people have used language and reading studies. They've studied drug use and craving studies. For example, with nicotine, they'll have smokers come in and look at pictures of cigarettes and different cues and see how this elicits the sort of the craving in the brain and the reward systems in the brain. And we also use emotional studies. For example, you might have different emotions come up, and you say, match the 2 that are the same emotion, or something similar to that. So when you design these studies, you want to generally alternate between 2 very similar cognitive states. So I think I talk about that a little bit more in the next slide. So when we also design these, we want something simple that a person can do in the scanner. So remember, the person has to lie still. So we don't want any head motion. So we don't want something that's going to cause the person to get excited and move around. We want to try to minimize their moving their head as much as possible. So that kind of limits sort of what you can do. We're also limited by the loud noise of the machine. It's hard to do auditory studies, although some people nowadays try to use those headphones that minimize noise and play sounds through those. Again, you have to consider that you have to use all like optical materials or things that don't use metal or electrical signals through metal wires in these. So when you design these studies also, you want to consider the designing and the power that you have in your study. So you generally have the person doing the same thing multiple times over the study so that you can put stack different things up and have enough power to do a statistical comparison. So if you just have a person do something once or twice and try to compare it to something else that occurred once or twice, you don't have a lot of power in your experiment. So the more the person does something over and over again, then the more statistical power you'll have. So we want to statistically see where the activation is, and we also want to keep track of the performance that the person is doing while we're in there. So we usually have a behavioral measure that we can keep track of to see how well the person was doing and to monitor that they're actually doing what they're supposed to be doing in there. So the behavioral measure is usually something that we can track, like a button press or an eye tracking event, or that we can see with the eye tracker. So like I said before, we want to design something that alternates between two cognitive states, but we want two similar cognitive states. So by doing this, we can see that we can take the areas that are activated similarly in both conditions, and during our statistical analysis, these areas will subtract out. So for example, if we're having finger tapping versus non-finger tapping, all the background things that are occurring besides just that finger tap will hopefully subtract out when we do the statistical analysis. So another example would be looking at faces compared to looking at blocks. So when we're looking at faces, we're activating a lot of different areas in our brain. We're activating the visual areas, we're activating this area called the fusiform face region, which usually becomes active as a person is looking at a face, and all the other areas that are associated with visual processing. But when we look at blocks, then we are also using these areas associated with visual processing. So when we compare the two cognitive states of this condition, all those background areas that were just used for the basic looking and processing vision are going to subtract out because we're keeping these two states related to each other. So hopefully in this case then, what we're actually going to see, the differences are going to be attributed to the blocks versus the faces, right? So we're not designing something that we're looking at faces versus hearing something, because then we're going to have the differences not only because due to the looking versus the hearing, but we're going to have all the systems that are involved with vision activated versus all the systems that are involved with auditory processing activated. So we're not going to be able to really pinpoint what measure we're going to see, because we're going to also see all this background activation. So when we design these experiments, we try to think of these things and try to only change one small thing so that when we see these differences in activation, we can say we're actually seeing a change due to this one thing that we varied between the two states. For example, reading sentences versus reading a non-sentence, so a bunch of words jarbled together. So in both cases, we're using the reading areas of the brain, the vision areas of the brain. The only thing that we've changed here is having something make sense versus having something not make sense. Another example would be somebody looking at violent pictures versus somebody looking at neutral pictures. So we've kept the basic background of both of these the same. All that we've changed in here is the nature of what they're looking at. Okay, so the activation maps then, after we do this statistical processing, are generated. So these are the maps that look at the differences between the two cognitive states. So again, we want the main areas of activation, for example, eliciting our visual system, to subtract out when we do the statistical analysis between the two conditions, and then only have the areas that are due to this slight change shown in activation. And here, this shows auditory activation versus visual activation. So in the example that I gave of having an auditory condition alternate with a visual condition, you would see that we not only get activation in something that maybe we were interested in, but we would get activation in all of these areas involved with the background systems. So I think you guys get the point. You try to minimize the differences as much as possible when you're doing the cognitive task. Okay, there's two different ways that we can design our fMRI experiments. The one is called a block design, and the second is called an event-related design. So in the early days of fMRI, most of the tasks were done in this block design. Nowadays, these event-related designs are more popular. We're going to talk about each of these a little bit. So when you do a block design, you have a person basically perform the task repeatedly and quickly over a block of time. So for example, we're going to see a study in a little bit, an example of looking at faces. So the person might fixate on a crosshair for a little while, and then they're going to look at a bunch of different faces that are coming. So they might see faces over and over again throughout this block of time. And the reason for doing that is because as the person is continuing to see these faces, the signal is going to rise in that area, and it's going to stay high while they're doing that. And then as soon as they go back to resting and looking at that crosshair, the signal is going to fall again. So the good thing about this technique is that we can get a good, strong, bold signal because we're repeating this over and over again. So we're assuming that we're activating this area that's looking at faces, and we're keeping this signal up high, and then when we turn it off, the bold signal is going to go back down to baseline. And when we do this, we generally allow enough space. So this picture here at the top is showing somebody looking at pictures versus looking at words. So the red, they're looking at pictures, they're looking at pictures, they're looking at pictures. Then they have a 10-second stop, and then they're looking at words. Another 10-second stop, and then they're looking at pictures again. And this break between the blocks is designed to allow the bold signal to go back down to baseline in the areas that we're activating and then turning off again.
So it is a set of pictures. It's not just one picture.
Correct. It's a set of pictures that they're looking at repeatedly over time, and that's the block design. So the advantages of a block design are they're simple and they're great for exploratory studies. You can get a really good, strong activation signal attributed to a single event because you're looking at these repeatedly, the signal's going up and it's staying high. We have a lot of experimental flexibility with this, and these types of studies are statistically powerful, so this is a good thing for us. Disadvantages might be that they're predictable and sometimes boring for a person. It may be difficult to sustain or control a cognitive design for a long time. The time response information is lost, okay? So because we're showing this repeatedly over and over again, we're actually not looking at the time event of the bold signal coming up and down again. Again, it's coming up and it's staying up throughout the whole period that we're having them do something, and then it's falling again. And another thing is that we're assuming when we do this that the bold signal is remaining constant throughout that block of time when it may actually not be. So those are some of the disadvantages of a block design. So response to these disadvantages, people came up with this other design called an event-related design. So an event-related design, you perform the task as a single event. So we're having the bold signal rise and then fall again. So for example, instead of looking at a lot of faces over a period of time, you'd look at a single face and then it turns off. So again, the bold signal, we're assuming that it's rising in the area that's activated due to this and then falling again after they're not looking at the face. So if you're going to do a design like this, you may need to allow enough time for the bold signal to return to baseline, which we said from the onset of that stimulus for it to rise and come back down takes about 15 seconds. So the time that we wait between these block designs is called the interstimulus interval. Okay, so this is a block design. This is shown in a slow experiment. It's a slow experiment because for each time we're having the person do something or having them look at the face, then we're waiting for the bold signal to go back down again before we show them another face. The problem with these types of experiments is that they're very slow. So if the person is only looking at a face once every 20 or 25 seconds, and they're all bundled up in a blanket in this little cozy, snug environment, they're going to start getting bored. They might have trouble staying awake. They might start thinking about other things. What am I going to have for lunch as soon as I get out of here? How much money am I actually making from the scan? Is it worth being in this scanner and being so bored? And these are also not very time efficient. So we're spending a lot of time in these experiments waiting for the bold signal to return to baseline. And because of this, we may have most of our time spent in the experiment waiting for this baseline to come back down rather than actually having the person actively doing something. Also, a disadvantage of this is the signal to noise is lower than in the block design experiments. So because of this, we can increase the number of trials per condition. So we could, again, this fact of keep having them do it over and over again to get better power statistically. But again, if we have to wait for the baseline, the bold signal to come back every time, this is going to be a long experiment for the participant. So people came up with these rapid event-related designs. So to do this, they shorten the interstimulus interval, and in this case here, we're showing this interstimulus interval is non-random. So we're having a stimulus occur every 3 seconds here. And the problem with this now is that we're having these bold signals that come up. You see in the next line here, this is a hemodynamic response. And when they see something related, for example, with this yellow stimulus, their bold signal is going to come up and come back down. 3 seconds later, they see a signal related, something related to this red stimulus. The bold signal also is coming up and down. 3 seconds later, the green stimulus, up and down again. And now we're starting to get this overlapping of bold signal. So this is also not a very good study design. We're not sure what's happening in the bold signal to which stimulus. How much is this rising due to the fact that we haven't let the signal fall all the way down yet? How much is actually occurring before the red stimulus there? It's sort of getting lost in the middle of the other two stimuli, right? So this isn't a very good design either. So people have thought a lot about this, and what do we do when we want to have these rapid events but still try to measure this time effect with this event-related stimulus? Well, the solution that they came up with for this is have a jittered interstimulus interval and a random stimulus presentation. So what this means is that instead of having this stimulus occur every 3 seconds, now they're going to have different amounts of time occur between these stimuli. And because of this, we have differential amounts of overlap in our bold signal. And one thing that this does is that it introduces some randomness, and it makes it a little easier for us to tease apart, because now we can see what the difference is here between going to the quickly from yellow to red versus the red by itself and the green, yellow, green. And it's easier to tease these things apart if we don't have a predictable interval between these. It introduces more randomness into the experiment, and it becomes easier to fit our heart rate functions to this. The HRF being, I'm sorry, it's not the heart rate, it's the hemodynamic response function, or the bold signal, okay? So this is a solution that people have come up with for these rapid event-related design experiments. So I think maybe we should stop right now and have a break. And then when we come back in half an hour, we can talk about some different types of study designs. And I'll show you some examples. So we're going to look at some example study designs. And these are some of the studies that we use in the experiments that we run at our center at the University of Pittsburgh. Some of these I've cleverly animated on here, and some I wasn't able to do as well. So we'll just go through these here, what I have. So a study that I used to use when we first started doing fMRI was called the Antisaccade Task. And this study is designed to measure cognitive inhibition. So a saccade is an eye movement. And so we're going to look at that first. Then we're going to look at FACES task, which uses implicit emotional processing, which I will explain to you. Implicit means it's a background process, so it's not an explicit process, it's an implicit, and you'll see how that works in a minute. And we're going to look at this thing that we call the reward task, or a guessing game task, that's designed to look at winning and losing in the brain. Okay, so the Antisaccade task is designed to activate both sensory motor and inhibitory areas. And the instructions that we give to the participants are fixate on the crosshair. Okay, if you see if the crosshair turn green, you're going to move your eyes horizontally towards the dot. If you see it turn red, you're going to move your eyes away from the dot in the opposite direction. So what we're going to first look at is the basic sensory motor task, which will be our alternative to this Antisaccade task. Okay, so in this first task, all you're going to do is look at the crosshair when it comes up, and as you see the green dots, you just move your eyes towards the dots. Okay, is everybody ready? Okay. Oops, I guess they're yellow, I'm sorry. You try to do this without moving your head. And of course, we pack the person's head in with pillows and try to keep their head as still as possible without giving them a headache from too much packing. Okay, so that was an easy one, right? Okay, the next one's the harder one, okay? So again, what's going to happen is if you see the crosshair turn green, you're going to look towards the dot that pops up. If you see it turn red, you're going to look exactly in the opposite location on the horizontal axis there. Okay, so there's our dot. Then we look towards it, right? So you're going to look to the other side, but it's very hard to do because the first thing you do is look at that dot. So that's the Antisaccade task. So we used to put kids in the scanner and tell them to do this, and they sometimes were successful, sometimes not so successful. You got better ones than others, and of course we record their eye movements so we can actually see which way that they're looking. And after all the tasks, whether they do a great job or not, we always tell them great job. We like to encourage them. And this is what our eye equipment looks like in the control room. And you see this this monitor here is labeled MR Unsafe. So things around the MR environment are labeled MR Safe or MR Unsafe. And this is the computer that we use. And we have this projector, like I mentioned, in the control room. And we also have the eye tracking system, which actually looks at their pupil and their corneal reflection. And this monitor system up here is actually, that's actually the person's eye. So we can actually see it pretty well and see if it's open or closed and see what it's moving around doing. The thing below it sort of is a better indicator to us as to whether their eye is moving back and forth, up and down. You can sort of track what they're looking at there. Okay, so do we want to have this a block design or event-related design? What do you think? Okay, so this is kind of a trick question because you could do it either way. Usually in our Saccade task where we just are looking at sensory motor, and we use that as a block. Or you could do it either way. See, on the top design, if we did it in blocks, we could do the pro-saccade, meaning that's when they're going to look at all the dots that follow. And then we could do anti-saccade for a little block of time. So this is the way we would do it. We might have them do 5 trials per block, or we could also do it event related. Like you said, we just have them do 1, then maybe switch to the anti, and the pro. And again, we would put different amounts of space between these, and we would make the events random. So we would randomly do whether it's a pro-saccade or anti-saccade so that the person didn't get predictable in what they were doing. Okay, so these are the areas that task would activate. So it does activate the visual areas. Interestingly enough, sometimes if it's a more intensive task than another, the eye areas do activate. So they tend to activate more in the anti-saccade task than in the pro-saccade task. We also see some of the supplementary eye fields and motor fields elicit for this. There's some areas up here called the frontal eye fields that activate to this task. And the big area that we like to see light up is this dorsolateral prefrontal cortex, dorsal and lateral. So that's an area sort of on the, this is a sagittal view on the right here. This is an axial view on the left, and the bottom is our coronal view. So if we were looking at these frontal eye fields in a coronal view, I mean, not the frontal eye fields, I'm sorry, the dorsolateral prefrontal cortex, we might see it around this area if we were looking at a slice further back. So these are the areas that activate, and again, this dorsolateral prefrontal cortex is often used and looked at in this task to see how well somebody's inhibition center is activating compared to somebody else's. And I looked at development in children in these areas and tried to understand what the differences were between children that were at high risk for developing drug and alcohol problems due to their biological parents, whether their parents were drug and alcoholics or not, drug addicts or alcoholics, and compared to these to control children. Excuse me. Okay, so now we're going to move on to the FACES task, and this is an implicit emotion processing. That means that we're looking at a process that they are not attending to. So they're not attending to the emotion in this process, they're attending to something else. And this is, when we do this, we're trying to look at the background emotional processing that goes on in the brain. So the instructions that we give them are pay attention to what you see and respond as quickly as possible. If you see a picture of a male, you press your middle finger. If you see a picture of a female, you press your index finger. So what they're attending here to is gender, not the emotions. So here's what the task might look like. Okay, so look at this dot, and then we're going to see some faces. And again, if you see a male, press the middle finger. Female, press your index finger. Okay. Okay, so that was our first block. This is in a block design. You could see those were angry faces, right? And interestingly enough, we try to get rid of all the other cues. So we just try to keep their facial expression in there. And sometimes people have a hard time telling male and female on some of these faces. Okay, so here would be our control condition. So in this case, you can see that the difference in our tasks here would be the looking at the anger emotion versus looking at a control figure, okay? So we're hoping to get the implicit emotional processing. So this person is processing emotions and they don't even know it. But in fact, some of these faces look a little comical and people think they're a little silly after they've been looking at them for a while. And the data set that those were created from were actually hired actors and actresses that were paid to make these emotional expressions of these faces. And it's a public data set that's available if you're interested. So the areas that light up in response to this task, again, are the visual areas. Like I mentioned, you'd think they would all subtract out, but they don't all subtract out. And we get really strong activation in the amygdala and emotional processing centers, which are located, again, in the limbic system in the midbrain. And in these, on the two slices on the right, this is the coronal slice in the middle and the axial slice on the right. You can see that really high activation. The red is where it's activated the most highly, and those are right where the amygdala are in the brain. So what you're looking at here is a group map. So we had a lot of subjects. We put them together statistically, and you can see that the amygdala lights up really nicely in this task. This task also activates the dorsal ACC, which I told you is that area of tissue that lies above the corpus callosum in the middle of the brain there. And it also activates the orbital frontal cortex, which is, I'll show you the spot. It's right here. And also you can see it here. And this area is thought to work together with the amygdala in the control and the regulation of what you're looking at in terms of emotion. Okay, so the last one I'm going to show you is the reward, or the guessing task. This one is designed to activate reward and loss centers in the brain. In this case, we do it for both anticipation and outcome. So anticipation would be they think they might win or lose, so they're waiting to see if they're going to or not. And outcome is when they actually find out if they win or lose. So when we first started doing this task, we used monetary amounts. So here's the instructions. When you see a question mark, you're going to guess if the number on the screen is going to be higher than 5 or lower than 5. Okay, the number is never going to be 5. It's always going to be 1 to 10. Okay, so actually 1 to 9. So it's never going to be 0. It's never going to be 10. It's going to be in between there. So you're going to make your guess, and then you're going to see the shuffling hand. So if you see the shuffling hand up, that means that if you guessed correctly, you're going to win. But if you guessed incorrectly, you're only going to break even. So you're not going to win or lose anything. Right, they guess when they see the question mark, and now they're guessed higher or lower. So they can press their index finger to guess lower and middle finger to guess higher. So it's a pretty simple, like the task that we have to use in the scanner, it has to be pretty simple. But if you see the shuffling hand down after you've made your guess, if you guessed incorrectly, you're going to lose money. If you guessed correctly, you're only going to break even. So you're not going to win or lose any money. And like I said, when we started this, we just did win $1 or lose 50 cents. Okay, so apparently people's loss areas are more sensitive to their win areas. So if we can make it a smaller amount for losing than it is for winning. Plus, we don't want the people to get too discouraged. So but what we do now, and my boss, she raised this up to you win $10 for a correct guess, or you lose $5 for an incorrect guess. So she's trying to up the stakes to see if we can get a better activation and people get more excited about more money. So after you see the shuffling hand up and down, you're going to see the actual number that it was, and you're going to find out if you win or lose. An up arrow of green means you win. A down arrow of red means you lose. And the yellow circle means that you broke even, that you didn't win or lose. And I tried to animate this, but it just turned into a giant mess, and I didn't know what was coming or going. So I just did it like this instead. Okay, so the guessing phase is on the far left. So they see this question mark, and then the instruction again. Guess if the number is going to be higher or lower than 5. So they make their guess and say now you follow the arrow up and they see the shuffling hand going up. Then they know now that if they guess correctly, they're going to win $10. If they guessed incorrectly, they're going to break even. So they see this hand, they're waiting. They see what the actual number was, and then if it was a correct guess, again, they win $10. If it was an incorrect guess, they just break even. Now, if they see the shuffling hand down, then they know that it could be it's going to be either a losing or a break even trial. So if they guessed correctly in this case, they're just going to break even and see the yellow circle. If they guessed incorrectly, they're going to lose $5. So this is designed, again, to tease out this anticipation phase. So they know if they're going to either win or break even, or they know if they're going to either lose or break even. And by doing this, then we can take the time period that was of the scans that were during the anticipation phase and look at those separately than those during the outcome phase. So we can tease this apart into looking at both anticipation and outcome. And so I suppose I should tell you that the reason that we can do this is because this task is completely fixed. So if it's a winning trial, it doesn't matter what they press, they're going to win. So if it's a losing trial, it doesn't matter what they press, they're going to lose. Of course, they don't know that ahead of time. We tell them that at the end when we debrief them. This task actually has some really strong activation. So I didn't show you the anticipation, but I'm showing you the outcomes here. And you can see, again, this is a group of a lot of people put together and how strong this winning effect is. We have the sagittal, the coronal, and the axial slices. And remember, I told you this activates the caudate very much, and you can see that here in the middle. It also activates the putamen, which you can see there. This is remember, those are the areas that are rich with the dopamine and reward receptors. So this actually works. It gets people excited in their dopamine channels. And interestingly, when they lose, they get a very similar response. However, we see a lot more negative activation on losing than we do on winning. So I haven't talked too much about deactivation. So I've talked about activation, the areas of the brain that are active when people are doing a task. But in fact, we can also see deactivation. So activation is when the bold signal goes up. Deactivation is when the bold signal goes down. And so for years, people were kind of thinking, what does deactivation mean? Nowadays, they think that it's an inhibitory process. So if you see deactivation in an area, they think that area is instead of being activated, it's being inhibited. So that area is being kept from activating. Does that make sense? It's sort of still sort of a gray area that we're not sure what exactly physiologically is happening. We know there's a change again in the bold signal ratio to see it drop instead of rise, But like I said, now the physiological explanation is this inhibitory process. So we also see this when we start looking at fear circuits. Initially, the fear circuits that are in the midbrain are highly activated, and then some of the frontal areas start lighting up, and they start inhibiting these areas and sort of shutting them down so that you don't get this strong fear response over and over again to the same stimulus. Okay, so we've looked at a number of things: history of fMRI, how it works, designing of experiments. Now we're going to look briefly at how to analyze the data, and we're going to do this in several steps. So first of all, when we design our experiment, we want to have good parameters. Okay, so when we're designing our experiment, we want to design it so we can get the best data possible when we get to our data analysis stage. So as I mentioned yesterday, when you sample in K-space, the center areas of K-space are the lower frequencies. The outer areas are higher frequencies. And this is a small signal effect that we see. So we like to get a nice, good low frequency signal so we can see the bulk of the image. But a lot of the changes that we see to fMRI are higher frequency changes, small, fine changes throughout the brain. So we want to make sure that we sample enough of K-space to get the best signal that we can. So in the left two columns here, on the left column here, is we see that we sampled all of K-space, and our pixel size was 1 millimeter, or our voxel size. Pixel, again, being the two-dimensional picture element, voxel being the three-dimensional. And you can see on the right side, this has only been sampled in low frequencies with a bigger voxel size. So again, the smaller your voxel size and the more of K-space that you sample, the better data that you're going to get. And again, you have to take the Fourier transform to get from the top the K-space to the bottom to the image space. So the first step in data analysis is basically processing the data to get it in a way that we can actually use it and run it through our programs, and to remove as much noise as possible, because again, the variations that we see over time in this signal, we want to be able to attribute to the actual physiological differences that are happening with the bold signal, and not something else that's going on that may corrupt our data and make us think that we see activation when we actually don't. Okay, so like I mentioned, the first step is to convert the images. Nowadays, the scanner does the Fourier transform for us. So the images that we actually get now are already Fourier transformed and in the form of brains already. However, the raw files that we get from the machines are called DICOM files. So these come directly from the scanner. DICOM stands for Digital Imaging and Communications in Medicine. And this is a very standard format for the images to come in. So they're standardized in order for everyone to handle, store, print, transmit them, so that everybody collecting medical data has a similar format when they get these images. And with working with these files over the years, I've learned that a lot of information is actually embedded in it looks like a string of numbers. I think I have some examples later when we look at an actual data set from start to finish, but you can actually get time and date information from some of the raw files just by looking at them, which is good to know in case you lose track of which data came from whom, which we don't typically do. We don't like to mix our data up. It's too expensive for that. But if we do, then we can go back to these DICOM files and get some information out of them. Okay, so the DICOM files include a file format and a network communications protocol. It's a little bit over my head. but that's what they claim is in these, and it was developed by the National Electrical Manufacturers Association. And like I mentioned, the time and the date are embedded in each of these files, and the time, by the time, I mean time down to the milliseconds is recorded in these. So the first thing we do after we get these DICOM files is put them into imaging files that our software can read and analyze. And there's several different types of imaging files that we use. Initially, we had what was called these analyzed file formats. And this was two separate files. So one data set would come in two separate files. One file would contain the header information, and the other would contain the image information. So the header information file was generally a pretty small file that you could read using your imaging software, and it tells you different things like how the data was collected, the direction that it was collected, the order in which the slices were collected, the time that it was collected. If you entered a participant ID number, it contains this information, while as the image information, the .img file, is basically the bulk of the image. So it's a very big file, but if you were going to try to read it by itself, you wouldn't have any of the information about that data in the file. So this is typically the way that fMRI data was stored for a long time. And nowadays we have what's called a NIfTY file, and the extension on these is .nii. And these files contain all the information in one. So instead of having the two separate files, now we have them all into one. There are other software packages. One is called AFNI. We'll look at software packages a little bit later, but not today, but another day. But these have also, some of these have also developed their own image types. So in the case of AFNI, they also have this two-file system where they have a header file, but their image file is called a .brick file. So because fMRI was so new, when we first all started in this, people developed their own file formats that could be read by their own programs. And even in Pittsburgh, we had this. We had these Pittsburgh imaging formats. But nowadays, as this technology has become more widely used, these things have become more standardized. And today, those NIfTY files, those .nii files, are the most commonly used types. Okay, so once we get our images into an actual thing that we can read into our software, the first thing that we often do is do this, what's called slice timing correction. So we only have to do this for event-related studies. We don't do this for block designs because we sort of smear all the time together in the block design and chunk it as one time. But in an event-related study, when we're interested in looking at the timing of the data, we do this slice timing correction. And the theory behind that is we collect the whole brain in 2 seconds, but it means we collect from a starting place back to the starting place again. And that means that over the entire 2 seconds, we're collecting this data. So not all the slices are collected at the same time. So what slice timing correction does is it takes all these differences in when the data was collected and moves them all back to the 2 second starting point. So because, like I said, each slice is collected at a slightly different time. And again, these slices are generally interleaved. So that means, like I told you, we collect every other slice, and then we collect every other slice. And this is, again, to allow our slices to get back to baseline before we collect the guy next to it, so we don't have this mix-up of signal influence in the different slices. And this kind of shows you again. So if we have a TR of 2 seconds and we have 6 slices, the first slice is collected at t equals 0, but the second slice is actually collected at 2.6 or 0.33 seconds. The third slice is at 4.6 or 0.67, and etc. So that when we get to slice 6, it's actually collected at 10 over 6 or 1.67 seconds so that we get back to our slice 1 at 2 seconds. So you can see how this time is sort of stagnated as we go along. And again, slice timing correction is just a technique that we use to pull all those slices back to the starting point of 2 seconds. So we can line them up and pretend like they were all collected at 2 seconds. Again, we don't need to worry about this step with block designs. The next thing, and this is a very important thing, is our motion correction. This is especially problematic with fMRI because we're looking at a series of images over time. So if we're trying to model one voxel in a piece of tissue over a period of time, and this person moves, so say the piece of tissue was here, and now the person moved, so we're not even looking at the same piece of tissue anymore. So if they move too much, this is going to really be a problem because we're going to see, especially at the boundaries of different types of tissues, because we're going to start seeing intensity changes over time to this person moving and not to the actual physiological effect that we're looking for. So motion correction is a very important step. So we're looking at intensity changes, hopefully due to this bold signal. So we don't want to get false activation due to motion. You can correct this for this during the scan and after the scan. So during the scan, a way to correct for this might be, like I mentioned, to pack this person's head in as tight as you can without having them feel really uncomfortable. And people have tried packing heads with other things other than pillows, because pillows also leave you a little bit of room for wiggle. But what they actually found is that if you try to put somebody's head in place with something hard and hold it into place, and you put them in there for an hour or more, it starts to become really uncomfortable. So they're trying to move around to adjust for being uncomfortable, and it actually can cause more motion, even if it's in there pretty tight. And of course, if the person's uncomfortable, they're going to start, having other things that are going on in their head, like pain or like maybe some anxiety, and we don't want that to happen. We want the brain to be activating due to our tasks and not due to uncomfortable things or other things that might complicate the results that we're seeing. And we can also correct for this after the scan, and this is what we typically do. And I think I show an example of how this can be done. But the question is how much motion can actually be tolerated. So you can correct for motion to a certain extent, but after the person moves, if they move too much, then even taking the data and trying to resample it back into the space that it was initially can not be effective in getting rid of these motion artifacts. And this is something that might be seen in a motion artifact case. And as you see, it really along the boundaries of the head is where you would start to see activation. And these green and blues are activation that was computed. And because this person was moving, at the boundary of the head is where we're going to see these intensity changes, not actually due to the bold signal, but because of the wiggling around. Okay, so to correct for motion, we take our time series of data. So along the x-axis of these is the image number. And what is done is that we estimate motion parameters there's a program that comes in and tries to estimate how much movement was done during the data by looking at slices over time and trying to minimize some kind of a measure. So it might be looking at intensity changes in the border of the head and trying to minimize those by jiggling these time images around until that parameter is minimized. So it's a sort of an optimization algorithm. And what we get then is we get motion estimates, and we get 6 dimensions of motion. We get the X, Y, and Z, and then we get the 3 rotation parameters. And this is an example. The top shows very little motion, and the bottom shows more motion. And you can see in the bottom image that we have a lot more blurriness at the edge of the image, especially down there at the bottom, than we do in the top image. And that's what happens with motion in fMRI. I think we look at that a little bit more when we get to actual data processing. So you can see more of how that's done if you're interested. So the next thing that we do is that we line up the person's anatomical scan with their functional scan. So this is something that we do so we can, again, when we calculate the activation, we can go back and put it onto the actual brain structure and see where it's coming from. So people have generally the same anatomy in their head, but everybody has variations. They have variations in how big their ventricles are. They have variations in the size of different lobes of the brain. And so we like to not make any assumptions about where we are on the brain, especially based on our low resolution functional scans. So we co-register these by lining them up using an optimization algorithm so that we can put them together with as little difference as possible, and then we can put our functional activation on top of the anatomical. So like I mentioned before, some people think it's very important to get this anatomical information first, because if we just get all the functional data but have no anatomical reference at the end, then we're sort of limited in what we can do with this scan. So a lot of times they'll get in there, we'll do the anatomical scan first, and then we move on to the functional scans. Okay, so after we pre-processed the data, got it into the image space and did some noise correction things, then the next thing we're going to do is normalize these brains into a common space. And this is kind of a weird thing to do, but when we want to study groups in fMRI and compare maybe a clinical group to a control group, the problem comes up that, like I mentioned before, everybody's brain has variations. So if everybody has a variation in their brain, how are we going to stack up brains of different sizes and different shapes to compare them? And this is kind of a problem because maybe somebody's activation that looks like it's in the same space may actually not be, because the brain has a different shape in that case. Again, there's variations in how much size of the CSF and contained in the ventricles, and things like that. So in order to be able to compare brains across groups, we have to normalize them to a standard brain template. And there's been a lot of efforts and work done in this area of trying to figure out what is the best way to normalize or warp everybody's brain into the same space in order to compare them. So when fMRI first started, everybody decided to use this brain. It was called the Taylor-Rack brain. And what it was is a book you can see here that was developed by Mr. Taylor-Rack and Mr. Tourneau. And what they did was they took a brain section, and they did this with actual brain tissue, and they cut the brain up and defined a coordinate system where each feature of the brain was located. So a stereotactic system is a 3D coordinate system, and again, this was developed by Taylor-Rack and Tourneau in 1967, and this was a standardized coordinate space designed for neurosurgeons. So he designed this, and then they wrote a book about it in 1988. So it wasn't too old of a reference system. They developed this atlas, and if you look at this book, each page is a part of this atlas. So each page can tell you exactly where different features and where different tissues were located in the brain. So originally, this was sort of the best thing that we had. So we all normalized our brains into this tail rack space. Again, this was originally used for surgical planning and for deep subcortical structures of the brain. They previously didn't have an axis of reference for surgeons, and they created this system. And it was based on the brain of a 69-year-old French female. And there were numerous stories about the preparation of this woman's brain for sectioning and all the time that they spent defining this space. So this is what maybe two different brains look like before and after normalizing to this tail rack space. So the brains on the left are in their original subject space. And then after normalization, you can see that they are now allegedly in the same space, and all the features are lined up so that if we were to put these two brains together and calculate some kind of group measure in them, all the tissues would be lined up correct at the same location. You can see that the tail racking brain sort of shortened the frontal lobe a little bit and sort of squished the brain up. So over time, people kind of started complaining about this tail rack brain. They said, well, this was based on the brain of a single cadaver, and it's based on the brain of a 69-year-old. So how about our younger people, and how about a living brain? The left and right hemisphere differences were ignored in this tail rack brain, and it was assumed that the whole brain was symmetric, left and right. The cerebellum was ignored largely in this, and some of the problems of this brain were notorious. For example, the occipital lobe of this woman was smaller than most brains were typically were. And this brain was also a European brain, so a French woman. So people said, hey, how about Asians? How about other cultures? How about African Americans? Were all supposed to take this standard brain and take all our brains and make them match this brain. So this was a problem. So they started thinking about how can we get a brain that sort of represents the general population. So next came along was this Montreal Neurological Institute brain. And this was created to be a more standardized representation of the general population. So what they did was they matched this approximately to the tail rack brain, but they kept they tried to make it, again, more representative of the general population. They took 240 normal brains and manually defined landmarks in each of these brains. So they took 241 different people. Then they took 305 normal brains and matched them also to a tail rack atlas. So they tried to line up all the features and the landmarks of these brains, and then they averaged these brains. So one of these brains has an average of 305 people. They also have an ICBM 152 brain. That's an average of 152 people. And they also took this guy, he was called Colin Holmes. They scanned his brain 27 different times and registered it each time to create a very detailed representation of a single subject brain. And these are the pictures here that I'm showing you. These are the M&I brains. And when you get an fMRI software package, usually these come in as a standard set of that package so that you can match to these brains. You can see on the left side there is the average of the 305 brains. On the right side there is the average of the 152 brains. And in the middle there is this guy, the Colin brain. He was scanned 27 times, which is the Colin 27 brain. And you can see how fuzzy the 305 is compared to the 27 of the Colin brain. But nowadays, these are basically the standardized brains that we use, and we warp people's brains into these spaces to try to line them up. Some people still use the tail rack brain today. So how do we do this? How do we take a brain and warp it into a normal space. So to do this, we do both linear and nonlinear transformations. So when we do the co-registration of a single subject's anatomical to their functional brain, we just generally do a linear transformation because we're collecting this data at the same time. So we assume that this person's brain is not going to warp or grow or shrink during the process of our single scan. But when it comes to normalizing to another person's brain, we may have to enlarge or shrink the brain a little bit. So the first thing that's done is it's generally moved around in a translation, which would be this way, up, down, forward, or backwards. And then it's rotated to try to fit it the best it can on top of this brain, on top of this MNI brain. Then the nonlinear part comes in, and they try to either zoom it or scale it to fit on top of the other brain. And then they take another step for nonlinearity in trying to shear it or skew it to make it match up to this brain. So there's several steps that go on in taking this single subject brain and warping it into this normalized space. Now, whether that's a good thing to do or a bad thing to do, we definitely have the trade-off of that. We're taking somebody's brain information and we're smearing it all around and resampling it. So that's probably not the best thing to do. But then on the other hand, how are we going to compare these groups of brains? Now another alternative would be to compute everybody's brain in a single space and try to anatomically segment so that you could pull out the activation, say, just in the amygdalas. But in that case, you would need to go through every person's brain and have somebody trace out where those amygdalas were. They're not so easy to pick out on somebody's anatomical brain. So a lot of times you have to get very close up to the tissue, so you have to blow it up a lot and try to make a subjective decision as to where the boundaries of those structures are. So the alternatives in both cases aren't that great. And of course, if you're going to do each person's brain like that separately, it takes a lot of time to do. The last thing that's typically done in our processing of our brain is called spatial smoothing. And this spatial smoothing blurs out residual anatomical differences in registration areas. So these slight anatomical differences that are occurring, and then we're warping them into a single space. Then we're going to spatially smooth this out so that we hope to sort of smear around those problems that we have and make them less starking. So we're going to spatially smooth. And what we do is use a Gaussian kernel, such as the one here, to blur the spatial information around. Now, I worked with a guy that just hated spatial smoothing. He thought it was like the worst thing to do to the data. You're trying to spatially localize where you're seeing activation, but you're smearing it all around first. So there's definite advantages and disadvantages to doing this. People will smooth these over between 4 to 8 voxels. So that's quite a bit of smoothing. If you think of each voxel as maybe 3 millimeters each, that's if you smooth over 12 voxels, I mean, you're really taking this information and smearing it around. So we do spatial smoothing, but we try to do it on a smaller scale just so that we don't distort the information too much. So each voxel is replaced by the weighted average of itself and its neighboring voxels in this case, okay? So here on the left would be no smoothing, and you can see if you spatially smooth too much, then you end up with just a big blurry image. Okay, so like I said, we could either do no spatial smoothing or we could do a lot of spatial smoothing. So the brain on the left has no smoothing. So you can see there's a lot of little spots in there, right? We're seeing activation in little pinpoints. And that might not be the best thing, as we're going to look at later when we look at data analysis, that sometimes these little spots of activation are just false positive areas, like regions of activation. So smoothing helps to get rid of those little tiny pinpoint spots. And at 4 millimeters smooth in the middle, you see that's not a bad compromise. This on the right here is 8 millimeters spatially smooth. And that really makes a very nice picture. It has nice smooth edges. It makes all our activation look like it comes in one big spot. And that's what people like about spatial smoothing. It makes their images look really nice. And with the popularity of fMRI increasing all the time, and different kinds of people from different backgrounds using this technique, you might choose to do a lot of spatial smoothing just so that your images look really nice without having a full understanding of what's happening to your data. So that's why this really has to be an interdisciplinary approach. And it's good if you're going to have something that uses such high technology as this to have a basic understanding of what you're doing in terms of processing to cause things that might cause problems or get results that are actually not accurate. So it's sort of a game between people who just want to use this and jump to conclusions, and on the other side, these highly technical people saying, how could you ever make that conclusion? You know, look what you're doing to your data. So it has to meet somewhere in the middle, I think. Spatial smoothing can cause reduction of spatial resolution, edge artifacts, merging of voxels, and mislocation of activation peaks. So that's it for the lecture for today. That was actually supposed to take 1 hour, but I see that it took 2, which is okay. I'd rather go through this slowly and have you guys all understand it. So then tomorrow or on Monday or maybe even Tuesday, we're not sure yet, but we'll find out. We're going to go on to start talking about more of the specifics of how this data is modeled after it's been collected, and we're going to go through those step by step. And it's general linear modeling. So when you come on in on the next lecture, get your thinking caps on. It's going to be one of those sort of data intense lectures again. So does anybody have any questions?